Good afternoon, everyone. Thank you very much for responding. You know how I like the call and response thing. So thank you very much for appeasing me. Um, doesn't it feel great to be warm? I walked in here, it was 2 degrees outside, and it's like 90 degrees in here. It's wonderful. All right. Um, I'm so excited, as I always am, when I host these events. But today we're celebrating the ninth annual Grad Slam at UCSF. Thank you. Appreciate that. So, as I just indicated, a particularly warm welcome, more ways than one, to everyone joining us here in the auditorium, but also to those who are joining us via our YouTube live stream. We're thrilled to have you here to celebrate these 10 extraordinary communicators. I'm Nikkei Blake. I'm the Dean of the Graduate Division and Vice Provost of Student Academic Affairs. And normally I would say I serve as your host today, but I'm sort of your co-host today. As we gather here in person on our campus in San Francisco, we want to acknowledge the Ramaytush Ohlone people who are the traditional custodians of this land. We pay our respects to the Ramaytush Ohlone elders, past, present, and future, who call this place, the land that UCSF sits upon, their home. We are proud to continue their tradition of coming together and growing as a community. We thank the Ramaytush Ohlone community for their stewardship and support, and we look forward to strengthening our ties as we continue our relationship of mutual respect and understanding. So Grad Slam is part of UCSF's celebration for National Graduate Student Appreciation Week. This is a national observance every year around the first week of April. And all week long, day number 3, we've been celebrating graduate students. So I do this every year so you know what's coming. If you're a graduate student, raise your hand. If you're a graduate student, keep your hands up. This is great. This is great. This is awesome. All right. Now, no, keep your hands up. Don't. I could have made you stand. I'm just asking you to put your hands up. All right. Now, if there's a graduate student in your vicinity, can you just turn around and give them a high five to the side? If you have to get up to give one, it's okay. Give them a high five. Tell them well done. Tell them we're so proud of you. Right. I love Grad Student Appreciation Week because, frankly, in my eyes, grad students are the engines of the car that we call research. You're the guys that do the hard work, do a lot of the hard work, not all of it, but you do a lot of the hard work. You have the creative ideas, and you come into this with such enthusiasm that we love to celebrate our grad students. So happy Grad Student Appreciation Week to all of you. All right, so if we did that, today we're here to celebrate our students, or extraordinary, talented communicators, and we're gonna have some fun. So if you don't like fun, it's okay to just get up and leave now. Okay, all right. I think you will agree that in these very fraught times, we need to grab any chance to celebrate. And while we may not have undergraduates or the traditional college sports, today's event is our own version of, let's call it, a college bowl. I used to say the Super Bowl, but then I realized that some grad students didn't know what the Super Bowl was. So it's all right. So we're going with the college bowl game, right? But make no mistake, this is serious business for those 10 guys. So before we go any further, I want all 10 of them to stand. And let's just applaud them for getting to this position. Are you feeling the love? Are you feeling the love? Yes? All right. All right. Just like in a college bowl, these brilliant graduate students will showcase their knowledge, creativity, and their communication. Each finalist has just 3 minutes to present their groundbreaking research, making every second count. We have a very well experienced timekeeper up here. He timekeeps for Berkeley. He timekeeps for San Francisco 49ers. So you will be kept on time. All right. For PhD students, the skills that they have acquired in preparing and practicing their talks today, and the poise they will have gained from this challenging communication experience will serve them well, no matter where their careers take them. It seems that every year I do this, I say that now more than ever, we have to learn to communicate. But now more than ever, we have to learn to communicate with all types of audiences. I understand that in the current environment with all the uncertainties and challenges, especially regarding funding of discovery science, it can feel a little daunting. However, I just want to take a moment to remind you of the incredible resilience, creativity, and passion that each of you bring to the research landscape. Your work is the cornerstone of innovation and progress, and even in the face of adversity, your dedication to pushing the boundaries of knowledge is what drives this institution forward. So remember, great discoveries often emerge from the most challenging times. Speaking of challenge, so I'm going rogue. I actually scripted it, but I'm going rogue a little bit. So how many of you are on TikTok? I love graduate students. They all act like they don't know what TikTok is. So it's all right. How many of you are on Instagram? Okay, so Instagram it is, no TikTok. All right, that's all right. How many of you follow the Grad Div Instagram page? All 10 of you should have your hands because all 10 of you did something for the Instagram page. Okay, so I'm sure this is not ingenious or anything, but there's a lot of misinformation, disinformation, weird information, truly terrible information on social media. And you guys have stories to tell. You guys got great stories to tell. How do I know this? I get to read some of your applications when you came in. And I'm telling you, I'm blown away every year. You have great stories to tell. So here's what I want to challenge you to do. I want to challenge you just to tell your story. Now, since half of you claim not to be on TikTok, it's all right. Don't go on TikTok, but that's where everybody is. Just so you know. Even I don't TikTok, but I stalk TikTokers, right? Even I do it, and I'm old, right? But seriously, I want to challenge you to tell your stories because a lot of what's lost right now is our stories. Now here's it. Nobody's on TikTok. Few people are on Instagram, maybe on Reddit, maybe on other things, X, Blue Sky, whatever it is. Yeah, okay. We have a new social media guy. Joey, I told you I was going to put you on blast. Stand up, turn around. If you've been following our Grad Flows, this is the guy that's responsible for it. Okay. Now here's what I want you to do. If you have a story that you really want to tell, come find Joey. We are going to put your stories on blast because I just need to see some positive things on TikTok and Instagram and Reddit. I guess that's not on Reddit, on X or whatever. So I want you to find Joey. If you don't know how to find Joey, find me. I'll hook you up. Okay? Okay, that's a challenge. All right. Now that I've taken half of Deanne's time, I'd like to introduce you to Dr. Dean Duncan, Assistant Dean. There you go. It happens every time. Assistant Dean of Graduate Student Affairs in the Graduate Division. I've enlisted, as I do every year, Dr. Dean's help because I know you love her so much, and she does a fantastic job to MC the program today. I just want to say you're going to see me walk out the room in a minute. I'm not bailing on you. I just got called into a meeting, so I'm going to run to the meeting and then try to run back. In the meantime, behave yourselves. Join me in welcoming Dr. Dean Duncan to the stage.
Hi, let's go. Let me get adjusted so I can get comfortable. How are you all doing today? No. We know better. How are you doing today? Okay. We're going to have a lot of fun. You know how I do. Sometimes I go off script as well, so I'm going to have a lot of fun. I have the right glasses on today, so I could start calling people out. I do that. You all have seen me do that. So let's have a good time. We're going to shout out the 10 folks in the back and have an experience today. So with that, today you'll hear from 10 of our close to 1,000 PhD students. PhD candidates entered the Grad Slam contest by submitting their talks on video, and a group of screening judges comprising faculty, staff, and alums reviewed the videos to choose these 10 finalists. These students all have been working very hard to make their talks as engaging as possible for you. So of course, the money at stake does not hurt. So for the competition, the grand prize is $4,000. That's rent, really, right? In San Francisco. The second place prize is $2,000. It helps. And then the third place prize is $1,000. So money all around, right? So plus you, the live and remote audience, will decide the People's Choice Award that is worth $750. You will have a few minutes to cast your vote online once all of the finalists have given their presentation. Those finalists who are not awarded one of the cash prizes will also receive a $100 Amazon gift card. So everybody gets money. Exactly. So the other way in-person audience can participate today is the trivia contest. And those of you who have seen me MC before, you know I like to shame. I gotta say it. Sometimes the questions are easy and sometimes they're hard. And if you get things wrong, I might start playing around a little bit, but we're gonna have a good time with the trivia contest. So between each speaker, I'll ask a question and the live audience member who gets it correct will also win a goodie bag from Campus Life Services. So everybody gets something today, and we're gonna have food in the back too. So everybody's a winner, exactly. So I know you all are anxious to hear students' talks, but before we get started with the presentations, I want to introduce you to our impressive panel of judges. So judges, as I introduce you, please stand. So the first is Lydia Bell. Lydia is the Assistant Director of the UCSF Center for Community Engagement, where she also collaborates on our Anchor Institution initiatives. Welcome. The next judge is Dr. Ishan Deshpande. Who is the Principal Scientist and Group Leader at Genentech, a UCSF postdoc alum and a UCSF Postdoc Slam champion in 2019. Let's go! Next, we have Dr. Alexandra Klein. Who is also a UCSF postdoc scholar conducting research on the effects of bodily signals on emotional behaviors. And she was also the UCSF Postdoc Slam champ in 2024. Let's go. We got another gem in our midst. Dr. Courtney McCormick. is a life science leader specializing in genomics and clinical operations. Courtney is a UCSF PhD and postdoc alum and the current president of the Graduate Division Alumni Association. Welcome. And lastly, Dr. Jennifer Nazarino. Jennifer is the Associate Dean of the Graduate Programs at UCSF and alum of the Sociology PhD program. Jennifer joined our team last July, and so for her, this is her first Grad Slam, so we have to welcome her. And this is a great opportunity for her to learn about this event and the work that you all do here as students. So thank you so much for being here and your careful consideration in choosing our Grad Slam prize winners this evening. All right, so the judges will be looking for the following basic qualities in their presentations. The first is, did the presentation help you understand the research? Second, was the topic and its significance communicating using language appropriate to an educated but non-specialist audience? And lastly, did the presentation want to make you know more about the research? Are you curious? Each talk will be timed, and any presentation that goes over 3 minutes will be disqualified. And as Dean Blake says, we have an official timer among us. You all know him as the UCSF Registrar, Doug Carlson. So Doug, as Dean Blake mentioned, is the official timekeeper for Cal football and basketball games. He spots down the downs and yards to go for scoreboard operate at 49ers games, and he operated the game clock for the NCAA West Regionals in 2022. So Doug is going to keep us on track, and he's going to keep me honest, too, with time. So thank you for being here, Doug. Okay, before we go into our presentations, I need one housekeeping. Please silence your cell phones. We don't want anything to distract the students as they deliver their talks, so no TikTok, Instagram. I don't even know all the things. Those are the couple things. No talking, texting, flash photography during the student presentations. Can we do that? Thank you. So judges, are we ready? Okay, finalists, are we ready? Audience, are we ready? So the finalists drew numbers to determine the order in which they'll speak, and our first speaker is Zach Kogan. Zach is in the Tetrad PhD program. His faculty advisor is Dr. Peter Walter, and the title of his talk is Lost in Translation, Dissecting the Molecular Mechanisms of Stress-Dependent Protein Synthesis.
The number one risk factor for heart disease, cancer, and neurodegeneration isn't obesity or smoking cigarettes. It's actually just how old you are, which makes you think if we could target the mechanisms that drive aging, maybe we could come up with new therapies for some of these horrible human diseases. Well, the problem with that idea is that we don't understand the mechanisms that drive aging. But that's exactly why my lab studies something called the integrated stress response, or the ISR. The ISR is a normal part of cellular health, but it gets dysregulated as you age, and it becomes hyperactivated in the brain. And if you inhibit the ISR in old mice, they start to behave like young mice in cognitive and memory tests. But the thing I really want you to understand about the ISR is that it is fundamentally a protein synthesis program, meaning that when it turns on, cells go from synthesizing one set of proteins to synthesizing a different set of proteins, and it's that shift that we need to understand at the molecular level if we want to target it therapeutically. So I performed an experiment that allowed me to ask for all 20,000 human genes, which ones are required for that shift in protein synthesis. And what I discovered is a factor called EIF3H, or the H subunit of EIF3. And when we remove that factor from cells, normal protein synthesis is totally fine. But the cells fail to shift into that ISR-dependent protein synthesis. Now, picture for a second that RNA is like a string that contains the information to build a protein, and that the ribosome is a bead that feeds over that string to build a protein. EIF3 is the hand that places that bead on the string. But the H subunit is like the fancy pinky sticking up, because we can remove it and that loading function is completely unaffected, which means that there's something else that EIF3 is doing during the ISR when the H subunit becomes essential. Now, what we think is happening is that some RNAs have tangles, and that during the ISR, the H subunit is untangling specific mRNAs. But what's most important is that if and when we can understand the molecular mechanism of EIF3H and of ISR-dependent protein synthesis, we can use that knowledge to selectively target stress-dependent protein synthesis, selectively inhibit the ISR, and in so doing, potentially create new medicines for age-related neurodegeneration, cognitive decline, or even memory loss itself. Thank you.
All right, Zach, thank you for starting us off. I like that pinky analogy. It actually reminded me, I don't even know what it's, I was thinking of something I used to play with as a child where it's on a string. So that, I just made that connection. So thank you. Okay, now, before we get to our next speaker, guess what time it is? Trivia, y'all know the drill. So it's time for our trivia contest. So for those of you, and I'm looking right at the camera because I know exactly where it is, those of you who are viewing our event remotely, during this time, it'll be a little bit like Jeopardy. In other words, there will be no prizes, but you can scream at the TV all day long, or your computer, or your phone, right? We hope you'll have fun guessing, right? So this is gonna be a fun time. So today's theme is potpourri. I know, right? I said the same thing. I was like, potpourri, hold on, the stuff that smells? Like, what you mean? And they explained it to me like I'll explain it to you. So it also means like it's watching Jeopardy where it's a bunch of stuff, right? So we're gonna have a mixed bag of questions that could be about science, pop culture, pop science. What's pop science? Do y'all know? Do y'all know what pop science is? What's it? It's Bill Nye? Oh. Thank you. I would think that's like pop culture, but thank you. San Francisco and beyond. So you'll find out what we're going to get into in a little bit. So, rules for our live audience. After I ask the question, please raise your hand if you think you know the answer. Obviously, no Googling on your cell phones, right? We don't have audience mics today, so Alexandra Simon, thank you. One of our Rosenberg Hill Fellows and one of our History of Health Science PhD students will point to the first raised hand that she sees, and if it's you, say the answer loudly, and then I'll repeat it for those at home. If you get the answer right, you'll get a goodie bag. And we want to thank Campus Life Services for generously donating the trivia bags, which include a $25 fast pay card good at numerous campus vendors, and plus some fun swag. So, once the question has been answered correctly, I will tell you additional fun facts. Okay? All right, so the first question is, the dress photo from 2015 went viral and revealed differences in how people perceive colors. What biological phenomenon explains why some people saw it as blue and black while others saw it as white and gold? Now I need, where is neuro? Where is the neuroscience? I don't actually have any hints. I just have the fun facts. We have a hand? Who has their hand up? Yes. So the answer that got called out is color blindness. No. Good try though. Appreciate you. Anyone else? Yes. Oh, you got real technical. So rod and cone distribution in between people. That actually is correct for a PhD. But let me go with the script. Right? So color constancy. So related facts. So that's the answer, but I agree with you. I studied visual neuroscience in my postdoc. So our brains try to filter out the perceived light source to determine the true color of an object. Besides, the lighting in the photo was ambiguous. Brains made different assumptions about the lighting, leading to different color perceptions. So, judges, let me check in with you all. Are you all okay for me to keep moving forward? Okay, I'm gonna keep moving forward. So I'm glad you're having fun with the trivia. So now for our next speaker, you ready? Okay. Now for our second speaker, Matt Arvidsson. Matt is a student in the Biomedical Sciences PhD program. His faculty advisor is James Gardner, and the title of his talk is Clearing the Air, How Cancer Hides from the Immune System.
So breathe me in and let me stay. Save my soul, don't make me wait.
Have you ever wondered how your immune system can respond to a virus that it's never seen before? The answer lies in complex machinery that generates immune cells with random specificity. In theory, these cells could recognize and respond to any possible combination of molecules. Now, this may sound great, but there's a big caveat here. Besides making cells that kill viruses and bacteria, you'll also make cells that target your own body. Everyone in the audience has generated immune cells designed to target and destroy their hearts, brains, lungs, and any other organ you can think of. Without control of these cells, devastating autoimmune disease is inevitable. Fortunately, there are safeguards in place. Before immune cells can enter circulation, they're screened very carefully by a specialized set of educator cells in a small organ called the thymus. The function of these educator cells is governed by one powerful gene called the autoimmune regulator, or AIR for short. AIR works as a sort of blueprint, enabling these educator cells to make proteins that are otherwise highly restricted to specific organs. They can then present these proteins to developing immune cells and test to see if they respond. If they don't, they're free to leave. But if they do, they're immediately killed. This concept is key to a term that scientists use called self. Self represents you, the proteins, cells, and organs that make up your body. The converse is non-self, viruses, bacteria, fungi. But what about something that lies in the middle? Something that is pretending to be self, but in reality is a corrupted, distorted version of self. That something is cancer. Cancer poses a unique challenge for our immune systems, and that's because they start out as our own cells. Despite growing out of control, they maintain many of the self-like features that they started with. This led us to wonder, could the same educator cells that protect us from autoimmunity be tricked into protecting cancer? We started by looking in tumors for cells that express AIR, the blueprint gene that controls immune education. And we found that every tumor we looked at in mice and humans, brain cancer, lung cancer, melanoma, and more, all had these cells. What's more, their abundance correlated with patient outcomes. The more of these cells, the worse the prognosis. Then we wanted to move past correlation. Could these cells directly contribute to tumor growth? To test this, we used special mice that enable us to delete all AIR-expressing cells. And when we did that, tumor growth slowed dramatically, and the immune system was immediately activated, suggesting that AIR is crucial for disguising the tumor. We're still working to understand how to target these cells in humans, but we hope that by removing AIR, we can choke the tumor and breathe easy again. Thank you.
I like your image, Matt. I like the creativity with the classroom. Very good. And the education, seriously, I like it. It matches for me. Okay, let's go for the second trivia question. Are we ready? In what country was the Caesar salad invented? Oh no, how do y'all know this? Hold on. Mexico? How you know that? Oh, okay. Yes, Mexico is the answer. So, some related facts. Caesar salad was invented in Tijuana, Mexico on July 4th, 1924 by Italian immigrant Cesar Cardini. Cardini created the dish at his restaurant Caesar's, which had become popular with American tourists during Prohibition. Oh, okay, this is the image. Oh, this is the second image. Okay. In its earlier days, the Caesar salad was often prepared tableside, adding a touch of theatrical flair to the dining experience. I know, it sounds good, right? The American chef and writer Julia Child, which is this image here, said that she has eaten a Caesar salad at Cardini's restaurant in her youth during the 1920s. She wrote, quote, one of my early remembrances of restaurant life was going to Tijuana in 1925 and 1926 with my parents, who were wildly excited that they should finally lunch at Caesar's restaurant. The salad was made with whole romaine lettuce leaves, which were meant to be lifted by the stem and eaten with the fingers, a dining experience she never forgot. I'm hungry. It sounds amazing, right? Judges, how are we doing? Keep going? Okay, thank you. Our third speaker, Katie Wedderburn Pugh, who is a student in the Biomedical Sciences PhD program and the Medical Scientist Training Program. Her faculty advisors are Drs. Tippy McKenzie and Elizabeth Crouch, and the title of her talk is Under Construction, Mapping the Blood-Brain Barrier's Blueprint in Development.
If you know Bob the Builder, you know he's built a house or two. But the brain has built a house of its own, and it's known as the blood-brain barrier. This barrier is made up of blood vessels with special cells and proteins, all carefully regulating what gets into the brain and what stays out. The problem is that many potentially life-saving drugs and therapies can't get past the front door. But the key to unlocking that door may lie in understanding how the house was built in the first place. The blood-brain barrier first appears early in development, and at these stages is, like a good chunk of UCSF's campus, under construction. By injecting a specific kind of dye into mice at these early ages and tracking how that dye gets into the brain, our lab and others have found these construction sites where new blood vessels are being built. In these sites, the barrier naturally allows certain things into the brain that it usually doesn't. We think that by understanding the blood vessels in these sites and how they differ from fully built blood vessels, we can design therapies that access the brain and be especially useful for diseases that need treatment before birth, when these sites might still be around. So what makes these construction sites so special? My research has found two critical differences in the blood-brain barrier as it's under construction. First, using a technique where we look at individual cells that make up the blood-brain barrier, a technique known as single-cell RNA sequencing, I found special types of cells that allow more things across the barrier. These cells are highly present in our construction sites. Second, blood vessels like to build on a foundation of a protein called collagen. And usually this collagen is sturdy, rigid, which makes sense when you're building a house. But by looking at slices of mouse brain at different ages, I found that in these sites, there's a different kind of collagen protein, one that's flexible and designed to bend and twist in ways that the typical protein simply can't. As we track these changes over time in the building blocks and the structural foundation of the blood-brain barrier, we're creating a blueprint that could change how we deliver things into the brain and potentially find similar areas in adult brains or neurodegenerative diseases. As we put together this blueprint, the real question becomes, can we build it? I think we can. Thanks.
I love your analogy too, Kaylee. Beautiful. Okay, our next trivia question. Whew, everybody ready to go? Which bone are babies born without? Woo, we had a hand pop up immediately. Yes. Yes, ma'am. Kneecap is the answer. How did you know that, Jessica? Things you read in your casual time. What looks like a kneecap in a baby is actually a piece of cartilage. It's not until later in childhood that this cartilage begins to harden into bone. Cartilage is more flexible than bone, easing the birth process and the transition the toddler makes while learning how to crawl to walk. The ossification process typically begins around 3 to 6 years of age, and the kneecap isn't fully formed until adolescence, after which it weathers a lifetime of use and abuse, which is true. And you may not be surprised to hear that the knee is one of the most stressed joints in the human body, relying on 10 different muscles of the hips and legs to stabilize it and help it function. So keep working those calves and quads. Make sure you go use the gym and lift weights to help out with your kneecaps. So thank you for that. Judges, are we ready to move forward, or we need a little bit more time? Okay. Our fourth speaker, Maggie Colton Cove, who is in the Pharmaceutical Science and Pharmacogenomics PhD program. Her faculty advisor is Dr. Hideo Akeda. The title of her talk is Building Biological Sleeper Agents to Fight Brain Tumors.
Bubble gum brain, my bubble gum brain, it numbs my pain. Bubble gum brain, my bubble gum brain.
The T cells of your immune system are your built-in security guards. They surveil your entire body, seeking out and destroying cancerous cells. Most of the time, they do a pretty good job. But as Matt just explained, sometimes cancer cells can slip by undetected. So we have a tool that helps us engineer T cells to be better at detecting cancer. We can take a patient's T cells and fit them out with a receptor that acts almost like an antenna, locking in on cancer-specific markers and directing the T cell's tumor-killing machinery against them. These receptors are called CARs, and CAR T cells have been incredibly effective against blood cancers like leukemia. But I work with a deadly subtype of pediatric brain tumor, and that's an entirely different story. The bloodstream is the natural habitat of the T cell. The brain, on the other hand, is more like a barren wasteland, difficult to get to and almost impossible to survive. Oxygen levels are lower, other immune cells that would typically boost the T cell's performance are absent, and the tumor itself sends all sorts of signals that weaken T cells. CAR T cells are incredibly activated, but activated T cells are also more likely to enter a state of exhaustion, where they can't perform their function anymore. CAR T cells are sprinters, but in the hostile environment of the brain, you need to be able to keep your pace. So my lab is working to improve CAR T cells for brain tumors by putting the control of the CAR under a genetic switch that detects a brain-specific protein and then turns on the CAR. I like to think of these cells as biological sleeper agents. When I inject them into the bloodstream of a mouse, they're completely normal T cells until they travel to the brain, hear their code phrase, and activate into tumor-killing machines. At first, the idea was just to make sure that these cells were only active in the brain and not targeting any other cells and other organs that just happen to look like tumor cells. But we quickly realized that by delaying the activation of the CAR, we were essentially allowing the cell to save its energy until it got to the brain. These inducible CAR T cells are stronger and less exhausted. They clear tumors faster and keep them gone longer. When I look at the brains of these mice weeks after injecting T cells, I can still see those inducible CAR T cells sticking around. These security guards are not sleeping behind the desk. They are alert and energized. My lab is currently working on a clinical trial to test these cells in adults with brain cancer, and I'm very hopeful that my research will help pave the way towards testing in pediatric patients as well. There is a huge need to find safer and more effective treatments for brain cancer. If we can harness and control the natural tumor-killing power of the T cell, we can bring new treatment options to the patients who need them the most. Thank you.
Thank you, Maggie. And I loved your analogy around the habitat between the immune system and the central nervous system. Beautiful. Okay, so I'm going to say this. Before I get to the question, I'm going to say this, and y'all not going to like it. I don't know if you're going to get this question. I'm just going to say it. So the question is, solve this mystery. Which writer holds the Guinness World Record for the most translated individual author? No, no. 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 These two here. Carla. No. No. Huh? Oh, ooh, you hear that? Let me pause. Hold on one second. So the hint is solve this mystery. So that's a clue to in terms of the genre. No. Who? Mm-mm. Yo! How do you know that? Did you look it up? So the answer is Agatha Christie. Did y'all know? Do you know who this is? Like real talk. You do? Oh. So hold on. What do y'all know about Agatha? I know I don't, that's not, okay, just let me ask. What do you all know? Can somebody just give me just a little? Say it again. Oh, okay. Oh, okay. I just need to get educated for myself too. Okay, thank you. So just a couple fun facts. Okay, I'm familiar with that. Okay, thank you. Known as the Queen of Crime and the Queen of Mystery, Christie was the author of 66 novels and 14 short story collections with 7,236 translations cataloged by UNESCO's Index Translationum. Judges, how are we doing? Okay, thank you. But I told you I was going to be hard, right? So I didn't lie. Thank you all for that engagement. I appreciate it. Okay, our next speaker. We ready in the back? Okay. Madeline Urbanek, who is in the Biomedical Sciences PhD program. Her faculty advisor is Catherine Cadwell, and the title of her talk is This Viral Astronaut is Mapping the Galaxy in Your Brain.
When we look up to the night sky and see the hundreds of billions of stars in our sprawling Milky Way, it's almost impossible to believe there are just as many neurons currently packed into each of our brains. And unlike the silent expanse of space, our brains are a constant hub of interstellar signal transfer flowing from neuron to neuron. These connections are fundamentally us. They encode our memories, our personalities, even whether we ourselves might have the courage to explore the frontiers of the cosmos. You can imagine then that it's really no small task for the developing human brain to wire together these circuits. Likewise, scientists have struggled to find tools that can simultaneously capture so much of the circuitry in a single experiment. This means we still don't know how a healthy human brain wires together, let alone how these circuits may differ in conditions like epilepsy or schizophrenia. What we need then is a brave molecular astronaut who is up to the task of mapping these constellations of connections within the galaxies of our brains. And my research focuses on training such a fleet in the form of rabies. You see, the thing is, rabies is already naturally inclined to map the connections made between our neurons. In a natural setting, rabies selectively infects neurons and then jumps from one to its upstream partner. And once it gets to that upstream partner, it rapidly replicates and sends even more rabies further upstream to the next neuron in the circuit. In this way, it exercises the disease-causing effects we may immediately think of when we hear the word rabies, a virus making its way in from an animal bite on the arm, jumping up to the brain, and causing damage all along the way. However, there are a few key changes we've made to our fleet of rabies. For one, we've taken away its ability to naturally infect cells. Instead, it needs a protein landing pad delivered by a second virus. In this way, we can selectively infect certain neurons or even circuits to streamline our expeditions. Secondly, we've given each rabies particle its own unique flag, which it plants in every neuron that it visits. Then, by mapping the trajectory of these flags across all of the connections a rabies particle travels, we can effectively reconstruct these circuits within a piece of human donor tissue. And using this technology, I've discovered that the developing human brain is a rich nebula of neuronal crosstalk, where mature neurons appear to be communicating with their more immature counterparts to build the permanent circuitry we use well into adulthood. And with this foundational knowledge, I am beyond excited to continue sending our rabies fleet to explore these galaxies in both health and disease. Thank you.
I like your analogy too, Madeline, with the viral astronaut and actually the Big Dipper connection. And I like Cassiopeia was my favorite as a child. So I love it. Okay, trivia question number 5. Which country has more pyramids than Egypt? Oh. Mm-hmm. No.
Right here, over in the back corner.
No.
All the way in the back standing. Antarctica.
No.
Right here in the gray long sleeve. Peru.
No.
All the way in the back is the blue long sleeve. Yeah, go ahead.
No.
Right here in the front.
No.
Go ahead.
Mm-mm. Somebody may need to go on their phone, even though I said don't go on your phone. What? Yeah, what's close to Egypt? No, we getting closer.
Back here in the white sweater. Go ahead.
Say it again, sir. All right now, Sudan. So Sudan, located directly south of Egypt, is home to nearly 200 pyramids built during the ancient Kushite kingdom starting in 751 BC. The area of the Nile River Valley where the pyramids are located is called Nubia, which was home to an advanced ancient civilization. I was going to give you one more. This one, I like this fact. While both were used as tombs, Sudan's pyramids differ in style. They tend to be steeper and smaller than the famous Egyptian pyramids. So we're learning something today. I'll tell you that much. Judges, how are we doing? Let me keep. So how are we doing? How are we feeling about the trivia? I'm going to ad-lib a little bit. How are we doing with the trivia? How are we feeling? Is it hard? Is it hard this year? They are? I thought so too. Every year I read the script and I'm like, yo, I don't know this stuff. And I'm fine with saying it. Like, I don't know it. So can we write this down? Like, can we ease up on the trivia? Okay, I'm gonna get ready. Judges, are we okay to move forward? All right. Speaker number 6 is Kingsley Chow. Kingsley is in the Pharmaceutical Science and Pharmacogenomics PhD program. His faculty advisor is Dr. Laura Van Veer, and the title of his talk is No One Left Behind, The Quest for Personalized Cancer Cures.
My life be like, ooh, yeah, ooh. My life be like, ooh, yeah, ooh.
We live in a world where nearly every aspect of our lives can be personalized. The clothes we wear fit our bodies. Our thermostats adjust to the perfect temperature. And hey, even our TikTok algorithms are finely tuned to our every scroll. So why shouldn't cancer treatment be just as personalized, designed to match the unique characteristics of each patient's tumor? You see, I work with I-SPY, the world's longest running adaptive breast cancer clinical trial to date. And I-SPY is special because it is not one size fits all. It is personal. Most clinical trials out there will just randomly sort their patients through different treatment arms, kind of like running a raffle for the right drug. Everybody gets a fair shot, but not everybody wins. I-SPY is different. It leverages a decade's worth of trial data to predict and match each patient to the drug that will most likely work for their specific tumor. And it works wonderfully. In fact, we've seen an increase in patients' response rates from 19% to 65%. That is someone's mother, sister, friend, getting a fighting chance at life. But what about the other 35%? The patients who still don't have a perfect match? The patients who deserve more? And that's where my research comes in. Before patients even begin treatment, I go in and collect a small piece of their tumor and then grow it in a specialized gel that mimics the conditions inside the patient's body. And this little tiny living copy of the tumor called an organoid can then be infinitely grown and expanded and be tested on. In fact, I tested a variety of different drugs on these organoids to figure out what works best for that specific patient. If they couldn't find a treatment on the trial, I will find it in the lab. But that's not all. I'm not just looking for treatments. I'm also looking for clues. I study organoids to look for biomarkers, biological signals that could tell me how a tumor behaves. So with these clues, I can then figure out what makes certain tumors more aggressive, why some evade therapy, and most importantly, how we can outsmart them. Every discovery brings us closer to a future where we'll be able to match the right patient to the right treatment at the right time, every single time. A future where no one gets left behind. Thank you.
I appreciate your pep in your step and your collar being popped, Kingsley, for breast cancer, so thank you. So our next trivia question, so I'm gonna be uplifting now. I think y'all are gonna get this, so Alex, I need you to be ready to get these hands because I think they're really gonna go up. So the question is, this Pulitzer Prize winner also scored a win at the Super Bowl this year. Who is it? Yes, Kendrick Lamar. So Lamar's 2017 Damn wasn't just a hit album. It made history. In 2018, it became the first hip-hop album to snag the Pulitzer Prize for music, proving hip-hop's artistic power. Lamar also broke new ground at the 2025 Super Bowl. He was the first solo hip-hop artist to headline the halftime show. His guests included SZA, Samuel L. Jackson, Serena Williams, and Mustard. Who watched it? Everybody watched it? Okay. You watch it again and again? On you? Okay. And so, what did I want to say? Also, his performance of Not Like Us signified a victory lap in the rapper's feud with Drake. I didn't actually realize there was a huge feud either, because I be doing really different stuff with my music. So I learn a lot of stuff from these trivia, even though I talk about it. Fellow Pulitzer Prize winner Toni Morrison also said of Lamar, quote, Kendrick Lamar understands and employs blues, jazz, and soul in his music, which makes it startling. His work is more than merely brilliant. It is magic. I know. That was a nice quote. Judges, am I okay to move forward? Okay. Our seventh speaker, Devin Schoen. Devin is in the bioengineering PhD program. Her faculty advisor is Dr. Melanie Morrison, and the title of her talk is Smarter Stimulation, Putting the pieces together.
It's electric. Boogie, woogie, woogie. You can't resist it. It's electric. Boogie, woogie, woogie. You can't do without it.
When we're performing brain surgery, I think we should be pretty sure it's going to work. For example, deep brain stimulation, or DBS, is a therapy that involves making openings in the skull and then inserting wires deep into your brain. This is a permanent implant that acts like a pacemaker, but for your brain. And it's a great therapy. It's been life-changing for hundreds of thousands of patients. But the catch is that it doesn't work for everyone, and we don't fully understand why. In fact, about a quarter of the Parkinson's patients who get this operation don't see any improvement. That means that if everyone in this room was getting a DBS implant, we would all get our hopes up, we would undergo this invasive brain surgery and all the risks and recovery that it entails, and then at the end, everyone in about the first 4 rows, you wouldn't get any better. And in fact, a few of you might even get worse. That's a really big deal. So what's happening? We know in general that a patient's brain health impacts how they're going to respond to this therapy. Already, the traditional markers, clinical markers of brain health, like how well someone responds to a medication, are driving our surgical decisions. Growing research shows that we can learn more complex facets of brain health from medical imaging. So we can think of all of these different aspects of brain health, whether they come from brain scans, from clinical tests, from patient histories, as individual puzzle pieces. And so far, researchers have been studying these single pieces in isolation. And no one has put all the pieces together to get a holistic view of how brain health impacts DBS response. And that's where my research comes in. UCSF is one of the largest DBS centers in the whole world. We implant more patients than pretty much anywhere else. And what that means is that I have access to really a massive amounts of data, or in other words, a whole mountain of these puzzle pieces. So I collect all of these brain health puzzle pieces, and I carefully set the puzzles so I can take a step back, I can look at the whole picture, and I can find important patterns. And then I can ask questions like, we have a new patient. Do they more closely resemble my good responder or my poor responder puzzles? And that's a really simplified way of saying that I'm using machine learning to build predictive tools, ones that doctors can use to determine how someone's going to respond to this therapy before they ever step foot into the operating room. We can tell who is unlikely to benefit from DBS, potentially saving those people from an unnecessary surgery and an unsuccessful implant. I mean, my poor responders, all of you in the first 4 rows, you would want to know that ahead of time, right? Because, yeah, when we're performing brain surgeries, we should be sure it's going to work. And my research makes sure that we are. Thank you.
Thank you, Devin. And you are very lucky to play electric slide and we didn't bust out an electric slide up here, okay? Because I was about to get ready. I love it. So thank you so much. Trivia question number 7. What type of music is believed by some to help plants grow better and faster? Jazz. Mm-mm. Yes, classical music. So some fun facts. So there is no scientific consensus on this question. But we don't know, right? But one notion popularized by experiments first conducted in the 1960s and 70s is that the vibrations from classical music can stimulate plant cells, promoting growth and development. I'm just gonna say this, I'm completely going off script. So I have a ton of plants in my apartment. I love plants. My team knows, because I'm always on them about the plants in the office. You need to touch the plants and talk to the plants. Sure, if you wanna play classical music, fine. But I'm telling you, at home, I'm touching my plants, talking about them, I'm saying, ooh, you're beautiful. Trust me, if you come to my house, you're gonna be like, whoa, Dr. Deanne, we had no idea. So just do what I say. I'm not even gonna go through the rest of this because my research is the right research, okay? You can play your classical music, you can play your Kendrick Lamar, you can play whatever you want. Just make sure you're touching and talking to your plants in a sweet way. Judges, are we okay to move forward? Okay. Before I move forward, hold on one moment, and I see that you're ready. How are we doing with the signs? You know, that's one of my most favorite parts of Grad Slam. Can I see the signs? Oh, can we get a picture? Can we get a picture of this, Joey? This is the stuff. Oh, I love it. Oh, look at Kaylee. Oh, so cute. I love it. Thank you. Oh, hold on. Keep it up. We need one more picture. Oh, the cats. Oh, for Maddie. I don't like cats, but I like that. I know I'm bogus. I don't care. I'm having fun. Okay, our next speaker, Sophia Miliotis. Who is in the Pharmaceutical Science and Pharmacogenetics PhD program. Her faculty advisor is Nadia Rowan, and the title of her talk is Finding HIV, a Swipe in the Right Direction.
I've been everywhere, man, looking for someone who can please me, love me all night long. I've been everywhere, man, looking for you.
5 years ago, after the worst breakup of my life, I did what any single millennial does. I got on Tinder. But if there's anything worse than heartbreak, it's getting trapped in an infinite scroll of tech bros, living in the cloud, but still down to earth. No matter how long I swiped, the feed never seemed to end. Funny enough, your immune system runs its own sort of dating algorithm, but instead of looking for love, it looks for a virus. Inside infected cells, major histocompatibility complex molecules, or MHCs, are like singles on a dating app. They're constantly swiping through tiny pieces of the virus, called peptides. If there's a match, MHC takes that peptide on a date to the surface of the cell, and the immune system can check in, like a skeptical best friend looking for red flags. And viral peptides are full of red flags. Once spotted, the whole cell gets destroyed. At least, that's how it's supposed to work. But some viruses don't play fair. Like HIV, the virus that causes AIDS. It evades immune detection by rapidly mutating, generating millions of unique peptides the immune system has never seen before. And as a result, some infected cells escape and linger in the body for decades. But despite being incredibly diverse, HIV peptides are still on Tinder. They're still matching with MHC. And if we could pinpoint the exact peptides that make it to the infected cell surface, we'd have a way to track down these lingering cells and destroy them for good. But here's the problem. Right now, we're simply guessing at which peptides might match with MHC and testing them one by one. And with millions of possibilities, we're stuck in that infinite scroll. My research changes that. I'm using a tool called EpiScan, which screens hundreds of thousands of HIV peptides at once. Here's how it works. I start with millions of engineered cells. Each one carries a single HIV peptide. Inside the cells, if there's a match, MHC heads to the cell surface. And the stronger the match, the more MHC shows up. So I can isolate the cells with the most MHC and check who MHC matched with. And that peptide may be the key to the next HIV therapy. Because finding love is a mess, but finding HIV doesn't have to be. Thank you.
So, Sophia, I needed this talk when I was a PhD student because I was in an immunology lab and I studied virology. I didn't understand virology. I was looking at the immune system, but this helped cue me right up. So thank you for that. So our next trivia question number 8. Y'all are gonna get this one. So before it became a hub for biomedical research and technology, Mission Bay was primarily known for what kind of industrial activity? No. Yes, rail yards and shipping facilities. I actually do hear when I'm in like Lyft rides, people are like, nothing was down there. It was like a wasteland, you know? And I'm like, oh, okay. So San Francisco's Mission Bay, once a marshy bay and estuary, transitioned into an industrial zone from the 1850s to the 20th century, featuring shipbuilding, butchery, and Southern Pacific Railroad yards. In fact, the designers of Mariposa Park, which opened across from the Mission Bay hospitals in 2016, close to Tidelands, for those of you who live there, designed the park's plaza, pass, and shade structures to echo the rail yards and railroad roundhouse tongue twister that once occupied the site. The current era began in 1999 with the groundbreaking for Genentech Hall, UCSF's first research building at Mission Bay, followed by Rock Hall, Rudder Center, and all the rest of the buildings that continue to pop up. Question for you all. I was watching a movie, not the other day, like a couple years ago, that featured this building. Do you all know what that movie is? I actually don't remember. So I'm asking you questions I don't know the answer to. Yeah, Tiffany. Which movie? Yes, it was Contagion, because I was watching it and I said, oh, that's Mission Bay. Thank you. Judges? Okay. Ninth speaker, Benjamin Sipes, who is in the bioengineering PhD program. His faculty advisor is Dr. Ashish Raj, and the title of his talk is The Brain as an Orchestra.
An orchestra has many instruments, but the music you hear feels unified as a single song. How does the brain bind together those various sensations into a single experience? This question is known as the binding problem, and it highlights a major mystery. We don't yet know the mechanism that allows the brain to both segregate processing into different brain regions, while also integrating that processing across multiple brain regions, all together to produce a unified experience. My research aims to identify this missing mechanism, and my work suggests that this mechanism may be critical to understanding how the brain changes across the lifespan. My approach uses a new area of neuroscience that studies brain harmonics. Brain harmonics are standing waves that live in the structural connections between different regions, but which help to organize brain function. If the brain were an orchestra and brain activity were its music, then brain harmonics are the instruments that play the music and give its unique sound. Now, previous work on brain harmonics generally views them as mathematical tools to simplify brain activity. But because they represent these waves that unite different regions together, I wondered, could they be those missing binding mechanisms? I thought that if they were these binding mechanisms, then they should form an integration to segregation spectrum in the brain. And because all of our brains must do this same binding, we should also all share the most integrative and segregative harmonics. Indeed, this is exactly what I found. The lowest frequency harmonics were integrative. They coordinated all brain regions together, like the steady drumbeat of an orchestra. Meanwhile, the highest frequency harmonics were segregative. They localized to sensory and emotional processing regions. They stood out like the soloists of an orchestra. I showed that we all share the same drummers and soloists in our brains. But in between these extremes was a third type of harmonic, the flexible harmonics. These were involved in complex and innovative cognition, like a jazz section of an orchestra. I showed that we each have a unique set of flexible harmonics, and that they change across the lifespan. Brain harmonics increase in flexibility during adolescence. They retain this high flexibility in adulthood, between ages 25 and 50, and then they gradually lose flexibility with aging. Together, brain harmonics may be that missing mechanism to solve the binding problem, and their relationship to aging gives them significant medical relevance. So the next time you marvel at a beautiful piece of music, consider that it may just be echoing the extraordinary orchestra playing inside your brain. Thank you.
Thank you, Benjamin. And I love instrumentals. So I'm wondering what's going on in my brain when I'm hearing my instrumentals rather than lyrics. So thank you for that. Our next trivia question is, I think y'all are gonna get this mainly because there are not too many options. So I know I was shady of me, but I said it. Which American pharmacy invented the chocolate malt milkshake?
All the way in the back, standing with the hat. Walgreens.
Yes, Walgreens. So Walgreens roots are deeply connected to the soda fountain. Early Walgreens stores featured soda fountains, which were social hubs and where they served treats like ice cream and milkshakes. In 1922, a Walgreens soda fountain employee named Ivar Pop Colson added a game-changing ingredient to malted milk, ice cream. Do we have a picture of him? No, right? Oh, okay. You can find one? Oh, okay. But by adding scoops of vanilla ice cream to the standard malt milk, he created the malted milkshake as we know it today. Was this good trivia question? Okay. Judges, we doing okay to move on? Or we need a little bit more time? Okay, a little bit more time. So nobody knows this because I just came up with it like today. So there's going to be a 10th trivia question. I didn't tell anyone on my team that I was doing this. So nobody knows. So after the last speaker, it has its own prize. I know. Get on the edge of your seats. Not only for the 10th speaker, but also for the last trivia question. So I'm going to move forward to our last and final speaker. I need the energy to be hyped. For Kai Trepka, who is in the Biomedical Sciences PhD program and Medical Scientist Training Program. His faculty advisor is Dr. Peter Turnbull, and the title of his talk is Gut Check, How Your Poop Can Hold the Key to Better Cancer Treatment.
I wipe my brow and I sweat my rust. I'm breathing in the chemicals.
Every patient is unique, yet most colon cancer patients receive exactly the same set of life-saving chemicals called chemotherapy. Despite decades of research in human genetics, we still don't fully understand why some patients take chemotherapy and feel fine, but other patients suffer debilitating side effects. Our human genes are only half the story. Living in our intestines is a community of trillions of bacteria called the gut microbiome. These bacteria come into contact with and chemically transform almost everything we eat, from food to pharmaceuticals. The microbiome varies from person to person, and maybe variation in the microbiome can help explain variation in chemotherapy side effects. I started my PhD studying how chemotherapy changes the microbiome. This means I spent my days working with poop. And while the smell is awful, the science is fascinating. I extract bacteria DNA from poop to measure which bacteria make up the microbiome in different patients taking chemotherapy. I found that when patients take their chemotherapy pills, their gut ecosystem is thrown into chaos. The bacteria that survive this chaos do something curious. They gobble up excess toxic chemotherapy and chemically transform it into an inactive byproduct, protecting themselves. Bacteria can protect themselves from chemotherapy, but can they protect us? To find out, I treated mice with chemotherapy in the presence and absence of chemotherapy-consuming gut bacteria. Amazingly, these special bacteria prevented side effects in mice. Moving back into patients, I found that I could predict certain side effects just by looking at the microbiome. In other words, some patients experience side effects because their gut bacteria are unable to clear excess toxic chemotherapy, while other patients are protected by their gut microbes. The microbiome is a diverse, complex ecosystem that helps make each of us into unique biochemical individuals. During my PhD, I discovered that chemotherapy-consuming gut bacteria can be used to predict, prevent, and maybe even treat chemotherapy toxicity, a first step towards a future where no patient is forced to choose between survival and quality of life. Thank you.
I'm really glad that you enjoy your research, Kai. I am. Okay, the last trivia question before we go into voting and taking a quick intermission while the judges deliberate. So this question is actually subjective, and I'm the judge of the answer. There's no Googling, there's no nothing, right? So the question is, we all like food here in San Francisco. I know many of you are foodies because we talk about it a lot. What is the best Burmese restaurant in San Francisco? Absolutely not. Boo! So it's Mandalay Restaurant is the best restaurant in San Francisco. Has anybody been there besides Zandrine? Oh, okay. So your prize, Zandrine, is lunch with me at Mandalay. So let me say this. Is there a student who had it right? I want us all to go. Is there a student who had it right? Okay, for real. That is my favorite. Oh, okay. That's smart. I like that. Who, Carla, you had your hand up too? I'm just picking people. Allison too? Everybody? Okay. Can somebody take some name down? We're gonna do something different. Alex, can you take some names down? So Zandrine, Allison, Carla, and what's your name? I can't. You gotta go. Just take your name down. What's your name? Yes, write it down. So I will follow up with you. We may not go to Mandalay. We may just order it in and do something on campus, okay? Okay. Thank you for that entertainment. I appreciate y'all. Okay, so before we go to intermission, I would like to invite all of the finalists back to the stage. Thank you all so much for both educating and entertaining us today. Congratulations. Big round of applause again, everyone. And you can return to your seats. Thank you. So, as the finalists are returning to their seats, just giving you a couple of housekeeping related to the voting today. So this is also our audience who's at home to play judge today. So everyone got a card when they came in. So vote once for your favorite talk to win the People's Choice Award by going to the URL or QR code that will be displayed on the screen momentarily. Those of you viewing remotely will also see this link right under the video in your browser or in the description area below the video on YouTube. You may need to refresh your browser. You can take a minute to think about it, but please submit your vote soon. We will only have the voting portal open for 5 minutes. And again, remember, just one vote per person. So the judges are gone. They left me. They're going to a top secret location to confer and decide on today's winners. So I ask that you take a short break, stretch your legs, see your fans in the back, get some pictures, and we'll return. We're behind. So just say 5:30. Come back at 5:30. 5:30-ish, okay? And we'll announce the winners. Oh, we're gonna do the code. There we go. So we'll see you in a little bit, okay? Come back around 5:30, 5:35. Okay, let's go ahead and get started. So welcome back, everyone. Thank you for taking the intermission and mingling and taking photos and giving hugs, etc. So before we announce the awardees, I want to congratulate again today's speakers on a fantastic job. So join me in welcoming them again. And so regardless of today's outcome, you all have done a great job and you should be proud of your accomplishment because I know how much practice you all have done. And I have also done like a 2-minute speech too. It's very difficult to do this. So kudos to you all on that. Before I go into the awardee announcements, I want to say a couple of thank yous. And so the script is written from the perspective of Dean Blake. So I'm going to say thank myself first. For emceeing the event. Thank you all for being a wonderful crowd per usual and having a good time with me. I want to thank our judges, both the screening judges and our final judges. This year's Grad Slam coach, Dr. David Blancha of the Office of Career and Professional Development, who had the students polish their talks. Thank you, David. The team from Educational Technology Services, who these dudes always get it right. So thank you. Campus Life Services for providing today's trivia prizes. And of course, our staff in the Graduate Division and Student Affairs for volunteering for today's event. And we want to make special thanks for our communications team who's been sitting up here. They're right over here, and our event specials team who's running around outside, and Elisa Bea in the Graduate Division who worked together to head up organizing for this event, from outreach to contestants, judges, and volunteers to collaborate on trivia questions to managing catering, handling promotion, and beyond. So this takes a team of folks. So please give a big round of applause for them. And lastly, a special thank you to the Chief Foundation for generously funding today's $4,000 top prize. Their commitment to supporting doctoral students in advancing and sharing their impactful research makes their contribution to this event especially meaningful. We are truly grateful for your support, and if you'd like to come up and say a few words, please do so. So thank you.
Thank you. I feel the energy, do you? It's getting hot. I want to thank, I promise this is going to be not 25 hours, it's going to be brief. First of all, I want to thank Dr. Blake for inviting us to this very special event. For the past 18 years, the Chief Foundation has been an advocate for campus research and publication in major research universities across the U.S. We encourage PhD students to use their knowledge, their creativity, and imagination to advance the science frontier. That's exactly what you do. Today we hear many new ideas and innovation. Congratulations. Let's continue. Let's not stop. Let's continue to contribute to humanity and make the world a better place. Thank you.
And I'll call you back up. Thank you very much. So speaking of prizes, we've come to the moment we've been waiting for. Time to announce our awardees. So I've asked the judges to come up. They know when they need to come up, and I'll also remind you to announce the prize winners. So up first, People's Choice. So the People's Choice Award has been chosen by both the in-person and remote audiences today. The student will get $750, and Jen will share the news.
All right, People's Choice Award. The winner is Sophia Miliotez.
we will announce the third prize winner chosen by the judges. This student will get $1,000. Gee, let's go. The third prize winner, Courtney, will share the news.
All right. And your third prize winner is Kaylee Wedderburn Pugh.
Yeah, you can. You can take a picture too. Okay. Second prize winner. This student will get $2,000, and Alexandra will share the news.
All right. Getting exciting. The second prize goes to somebody who explained a very complicated topic very clearly, to Maggie Colton Cove.
Okay, the moment we've been waiting for. Now we'll announce the first prize winner. This student will get $4,000, and the UCSF winner will also go to compete in the UC system-wide contest on April 29th. Oh, I'm going to be out of town for a chance to win as much as $6,000 more. So, yo, rolling in some serious dough. So presenting this award will be Sam Chi of the Chi Foundation. So you can go ahead and open it and read the name.
Before I announce the name, can I ask everybody to stand up?
Yeah.
The first prize winner is Sophia Maniatis.
Okay, well, so congratulations to you all of our winners and all of our speakers today. Thank you for a huge thank you to all of the finalists for all the effort they put into their task. Great job. And thanks to everyone who joined us today in person and online. This concludes our UCSF Grad Slam 2025 event. One thing I will say, again, Dean Blake had mentioned this in her openings. Right now we're in a really trying times, and what I've really been communicating with students is to stay in community, stay grounded, and stay informed. If you need anything, please know the staff in the graduate division, we're always here to support you, as well as all the colleagues in this space, okay? So stay connected. And with that, have a great night and enjoy the rest of Grad Student Appreciation Week.